Okay, perfect. And everyone disappeared. Okay. All right. So I'm just gonna present this in an informal way. So please go ahead and interrupt me if you have questions or want to talk about anything. I'm gonna talk to you about the instrument and kind of how it's being used on the coastal side in California and what's going on that end. And then I figure we can talk, as a group about freshwater or estuarine applications after kind of getting a sense of the instrument. So right here we have the imaging flow Cytobot, which is it's outside of its housing, but it's a long tube that's about as tall as my 1-year-old son, so about 2.5 feet. And it can be deployed in a number of different ways. Um, and so, why do people care about this? Why monitor phytoplankton community composition? And so, I think for this group, that's obvious, right? You wanna look at who is in the phytoplankton community, and you can start to see, oh, a harmful taxa is starting to dominate the community. Therefore, we are at an increasing risk for a harmful algal bloom, right? So how do you do that? So in the marine context, so again, I'm going to kind of give this in a marine context, and then we can kind of draw parallels into freshwater. But on the marine side, one of our big harmful taxa is called Pseudonychia. It makes domoic acid, which causes all kinds of neurological effects for humans and wildlife that are exposed. You might have seen in the news like the sea lion stranding events and things like that that happen in relation to this type of harmful algal bloom when it happens along our coast. So this is the main application of the IFCD in California's marine waters is looking for harmful taxa, one of the primary targets being Pseudonychia, and looking for increases in abundance that can trigger things like additional sampling or send out early warning notifications, etc. Right? But assessing community composition is difficult. So it oftentimes this is a grab sample that you're doing. It gives you a momentary snapshot of, okay, at this point in time, this is what the phytoplankton community looks like. You usually need to bring it back to a laboratory. And then if you want any kind of type of quantitative count with a number associated, that is, a lot of extra time to do a settled count or some sort of quantitative microscopy count. If you want to know about more than one thing in the sample, then you need somebody that has training in taxonomy. And I will say, on the marine side, we say that, and I know that on the freshwater side, it's even more complicated to do the counts and um, the IDs because the way that cyanobacteria are than even in the marine system. But that's, it's an addition, it's same similar challenge in marine hab systems. And so, just to give you a sense on the marine side, in case you're not familiar, this is kind of the basic water monitoring program that exists. There's a separate public health program that I'm not going to talk about, but that does all of the mussels testing, and that's a collaborative program between California Department of Public Health, Fish and Wildlife, and OEHA. This is additional monitoring that has been funded through federal sources for the last almost 20 years. So in the early 2000s, a program called HabMap or Harmful Algal Bloom Mapping and Alert Program was developed. And so what this did was take a lot of the researchers that were working on marine habs in isolation and try to coordinate them in a programmatic way to use similar methods at their peer and kind of peer-based or very near shore monitoring stations. I'm showing that map here of where the observing is happening. And so that has now been going for since 2008, so a very long time for a monitoring program. And it's been a great program where there's weekly peer monitoring. They do water grab samples. One of the main things that they do is a phytoplankton community composition analysis. So they do a qualitative one, so similar to what On the cyanobacteria side, you do like a grab, and then you look at what the most dominant and subdominant are, and then a quantitative microscopy count that comes shortly after that that gets blasted out on an email listserv to those that are interested by all of these locations up and down the coast, and that happens on a weekly basis. And it's funded by the Ocean Observing Network, which is a federal NOAA funded program. And so there's been a lot of work over the last almost 20 years on, using this. There's publications and a lot that's been learned. But it suffers from the problems that I just mentioned where, you get your one snapshot of what the halves look like that day. And then oftentimes, my PhD, I worked at the Newport Pier Station. You'd come back the week later and you're like, there was nothing here. And now there's a big bloom, right? And so that's where, um, why am I bringing this up? It relates to the IFCB for a couple reasons. So, the IFCB is a type of instrument. It's an autonomous instrument that you can deploy in the environment. It's automated. You can submerge it, and it essentially will, in a semi-continuous way, sample the water and analyze the water, pulling a sample once every about 25 minutes, depending on how you set it up. You can do longer or shorter. And it has the capacity to be telemetered, to send the data somewhere, and you can automate the identification. And so it's a really powerful tool that, you go from, on the marine side, you go from, our weekly sampling system to, if you were to create a network, which we have, at most of those locations, you deploy an IFCB, and then you now all of a sudden have like a more continuous stream. I say that like that's a really easy thing. There's obviously like a lot that is under the hood. But just to give you a sense of how this is being applied in California, there was the IFCB network, which was envisioned. It was a project led by Clarissa Anderson at Southern California Ocean Observing System to establish this, and I was one of the PIs on that in 2021, where Ocean Protection Council funded the purchase of several of these instruments that are then kind of strategically deployed at many of those HabMap stations. So we're able to leverage the weekly visit to kind of check on the instrument, make sure it's working, but also complement some in situ grab sampling with this kind of more continuous data stream. And so there's that, and then there's a couple IFCBs that are just floating around that get, pun intended, that get put on moorings and that go on. There's a couple different cruise efforts that happen where people do transects on a monthly or by quarterly basis, to get more offshore information about phytoplankton community composition as well. So it's a really ambitious network, and there's kind of a lot that goes into just running a network like this. So I'll talk about that, but I know a lot of you are really excited to just hear about the IFCB as well and just the instrument itself. So here's kind of like the basics of how the instrument works, right? So this is the instrument in the center here inside of its housing, right on the top. I don't know if you guys can see my mouse when I hover, but we'll do kind of like a what it looks like when you take it apart. But it essentially can pull a sample into the instrument through a fluidic system. The cells are pulled into a fluid stream, so it's basically a flow cytometer, if you're familiar with that. So it has a fluid stream and it'll pull the cells and kind of line them up into a single file line to then run in front of a sensor, which has a laser beam that you, depending on how you want to set the instrument, can be activated by chlorophyll A, which is normally how we activate it. So it's like, okay, if it's phytoplankton, the sensor will trigger, um, uh, and then that will trigger a camera that's in there, a camera assembly to take an image. And then it has an onboard computer, which essentially assembles all of those images into a single file, which are then sucked up into the cloud and sent to a server, to be available to view. So this is just kind of how some of those images look in the background here. But you can also have automated cell identification, which is which is great, but it's also a pretty arduous process to get that to work. Um, so the instrument itself, when you take the housing off, it has a lot going on. So these are challenging instruments to maintain, but it essentially has a pump here behind this assembly, and then it has a syringe, right? And so that syringe is a 5 ml syringe. That's why you're taking a 5 ml sample. And then it runs for about 25 minutes to run through all of the water that's inside of that syringe. But it's fun and exciting when you take the housing off and it pulls the motor pulls the syringe back and you can see the water go through. Um, and then on the cell detection kind of pieces, it has a laser module and a power harness down here, which has all of the electrical assemblies to essentially run the laser. And then it has some ability. These are on the right-hand side. There's laser adjustment modules. So they are able to be operated remotely if you want. So if it's in the housing, you can operate and adjust the visualization through the way the laser um, is oriented. You can also do it manually if you've taken the housing off, but that essentially kind of fine tunes and makes sure it's catching all of the phytoplankton as they pass through the laser path. And the reason you'd want to be able to adjust it from a distance is a lot of these are deployed on piers. So say it gets hit by a wave or something, it can kind of put the instrument out of alignment. So you can try to realign it from back at your desk without going into the field. That doesn't always work, but at least, you can try and then you're like, okay, I didn't get it. Now I got to go out and pull the instrument and take it out of its housing and readjust it. But it does have some stop gaps that sometimes it saves you, depending on how active the environment is. Um, and then this is the imaging assembly. So it has uh, two different laser options. Essentially, what we call the PMTB, which is this is the laser that would activate chlorophyll. You can also do it as just a light scatter if you're interested in things outside of the fight, just something containing chlorophyll. So if you just wanted to count all the particles, you could do that. It actually, a lot of people don't use this because it increases your data input significantly, which can slow the whole system down. It has a detector assembly and a lot of stuff kind of down here. So operates a lot like a microscope where you align the iris. You have a filter and a diffuser that you align, your condenser and your objective. So all of this is kind of when you do any maintenance, a lot of it is like alignment, and that can be a lot to get the instrument all aligned and have everything in the line so you can get the images and be capturing the images so that they are suitable for identification down the road. Um, a lot of these parts do have the ability, not all of them, but a lot of them have the ability to make some adjustments. They're motorized. They have like a motor and an automated like motor type assembly that you can do from your back, at home base when you talk to the instrument from a remote location, and then they have a manual option as well. And then you have the onboard kind of computer and other fluidics control. So it has a whole essentially dual core single board computer at the bottom that runs everything and allows it to connect to the internet and allows you to control the instrument from a different location if you have it connected to the internet. Um, you can also, if you're not connected to the internet, you can set it to run, and then if you wanted to talk to it but not take the housing off, there's an assembly on the top essentially that you could connect and talk to it as well. And then it just has several motors here to control the syringe and control all of those different abilities to make fine-tuned adjustments from remote location. So that's kind of the instrument itself. I hope that you're just taking away by looking at this that there's just like a lot of pieces to manage, and so they're very powerful, but they're very intensive, and there's like a 3-day training course to learn how to run the thing, to learn how to run all of these different things and really fine tune it. So the people that run it are very, um, they do a lot, and there's a lot to kind of keeping them going. Um, they're not like a plug and chug, like plug it in and let it go. Um, but the IOCD itself, you can use it in a couple different ways. So you can use it in what we call like the benchtop mode, where you can set it up in the lab and essentially give it a sample that it sips from, and it can take the images within the lab, and you can plug in a screen and see like the phytoplankton community composition as it's counting. And then you could pull that off and run it through an automated classifier if you want. And then there's the deployed method, which I think is where the instrument really shines, is it can be deployed in the environment. So I'm showing a picture here where this is the IFCB on this picture. This underwater picture where it is hooked up next to some other kind of sensors. And so there's like a chlorophyll, temperature, salinity sensor, and then also sipping and taking the phytoplankton measurements at the same time underwater. And they can be deployed like that for several months before they need maintenance, assuming nothing happens to them. So no big storms or, things like that. So we've, as a network, come across, we've had oil spills, we've had storms, we've had a few other kind of extreme events where you have to be aware of those. And so for example, we had an oil spill in Orange County a couple years ago while we had the IOCB out, and we're like, okay, we got to turn the thing off. So you just turned it off and kind of weathered the storm so you weren't sucking up oil, and then turned it back on when everything kind of cleared. So, but we were able to do that from a remote location. So if you have something like this and you're deployed, it's not like a set it and forget it. You do have to kind of pay attention to what's going on in the local conditions. So your instrument, like on a big storm, you'll also want to turn it off so that if it floods when it turns on, when it's on, it's kind of that's a major catastrophic. If it's off, there's like salvageable things. So you make decisions based on conditions about if you're going to keep it running or not. You can also take it on research vessels. So here it is on a research vessel where it's essentially research. A lot of research vessels kind of pull water as they're cruising. So they do this in San Francisco Bay, for example. They have done this for many years where they sip off and get kind of an underway distribution of the phytoplankton as it samples the underway system as the cruise is ongoing. So that's a really common way that people use this as well. So as far as the IFCB network for California goes, there's several pieces. So I think the goal at the end here is to have the network deployed and running as much as possible for, barring things, acts of God kind of things that stop the instrument, but have them running as consistently as possible. And then you just wake up in the morning and you're like, oh, I have an email and it says that there's an increase in pseudonychia that's happening. So I didn't even have to go outside and I know this is happening, right? And so automated hab alerts is kind of where we're trying to get the system. But there's many steps that lead up to that. Um, the first two, I'll kind of talk to you a lot of like the instrument setup and maintenance. So I just kind of gave you a sense of like how the instrument works, right? So there's a lot that goes into setting up the instruments and the quality control. And then how do you manage the real-time data? Because it's a large data stream. It's image data. Um, so the IOCB network, I think some of the things that we're really proud of is the standard operating procedures. So setting up the instruments in all of these different orientations. Here's the mooring. Here's the IOCB on the Newport Pier at Stearns Wharf. They have it set up where it's in a little hut that's on the pier that has tubing that goes down and sucks water back up because you set it up in different ways depending on your location. Um, here's Cal Poly Humboldt where it's on up top side, but on a pier with a power box here. So setting them up in these different orientations, but then making sure that they're kind of doing the same thing. So there's a lot of ways that we have worked to ensure we have data quality and comparable data. So we have a standard operating procedure where we've kind of done experiments to look at try to normalize that we're sort of counting and have the lasers set in the same way. So you're counting at the same thresholds, for example. And then there's the network. We have monthly technical meetings, and those are great. I think, frankly, they're equal parts therapy for the operator as they are, troubleshooting and getting through different issues and learning from each other. How do you deal when it's really turbid? How do you deal when you have an oil spill? How do you deal when you have storms? And learning a lot from all the different operators. So then on the data management time or side, there right now, I have the link down here in the bottom. Um, but basically, there's an online dashboard that you could go to if you type that in. You can go to it and you can see all of the different instruments and all of the data that is available. So SCUS has a server essentially that's hosting all of this information and presenting it back out. This is they come out, you get the images and we present them in mosaics. So in this dashboard, you're just getting the phytoplankton images. If you know your taxonomy, then you're like, okay, I know what I'm looking at. If you don't, then you're like, oh, pretty pictures. But that's, the first kind of pass at making the data available and public across the network. Um, then the next piece, right, is okay, say I'm not a phytoplankton taxonomy expert and that image was beautiful, but it doesn't mean anything to me as far as what is the hab risk. Then we get into how the real-time image classification works and how do you present that information. And this is a lot of the current work slash kind of moving into that automated alert space. But the image classification piece is really interesting. This is a part that I worked on a lot. But essentially, we're building image libraries for every phytoplankton taxa that you want to identify. And you also have to build libraries for other things that you need to put into a category because the way that the algorithm works is it needs to be able to take images and put things into bins and then assign like an identity. So what this really means is a bunch of people for a couple years spent time in all of our copious free time taking images and being like, this is a seratium, this is a seratium, this is a seratium, and putting images into bins so that the neural network could then have, a couple hundred examples of the different bins of what that particular organism would look like in order to then make the automated assignment, right? So you have to give it an image library where somebody has assigned an identity. It goes into your neural network, AI kind of image classification. It's not really AI, it's more of a guided like um, neural network model that then places everything into categories, and then magic. Uh, you get classified data. I have a typo there, not data outputs where you can put things into categories. So this is a plot from a couple years ago where we were really focused on Lingulidinium, polyhedra, and then everything else we kind of binned into the higher level um, taxonomy, but you can track different taxa over time that you're interested in, and look at, is it increasing, is it decreasing, and start to get nice images like this where you learn about the temporal trends in a given taxa that is being identified. And so there's been a lot of work on that model, and so now there is a synthesized harmful algal bloom dashboard for this network where the data is being presented. It is in pilot mode, so I looked at it yesterday, and there were a couple places where it was throwing an error. But you can essentially, the goal is that this now gives you, you can look at a given location and then you can look at different classified images, and it gives you kind of some readouts on, we saw an increase, we saw a decrease in whatever taxa you might be interested in, and you can kind of look at outputs in various. You can look at carbon concentration or actual cell abundance. um, and then the data is available as a downloadable form. So it's this actually released to the public technically, but definitely in pilot mode still. So what we're really working on now is automated image alerts. And so we're trying to refine the classifier to make sure that it's classifying things as it should be, and trying to translate them into meaningful numeric outputs. So there's challenges with marine HABs, just like there are with freshwater, where, for example, an actual count, especially for something like pseudonychia, which I should have shown you a picture. I need to not assume that everybody knows what that looks like, but it's a chain forming organism. And so doing counts of chain formers is similar to colony formers, so like microcystis, where you're like, how many actual cells is that versus how many chains, and how are you then translating that into a meaningful risk threshold, and how are you sending out notifications in a way that is meaningful versus getting 15 emails a day from the ISCB telling you, oh, it's up, it's down, it's up, it's down. So we're working through kind of those logistics pieces right now. Um, and so I'll stop, but that's kind of how we're applying the imaging flow cytobot right now. And I say we because this is a huge effort from like, a couple dozen people. Special shout out to a lot of the staff at SCOOS. They're really on the forefront of this. Clarissa Anderson and Kasha Krenzen in particular have done like a ton of work on the classifier and just getting the network in place. So this is not work in isolation by any means. So I'll pause and just see if you guys have any questions or want to talk about what are the parallels or how would this work in a freshwater system?
Thanks, Jamie. Before I start asking questions, I want to let it somebody else, if anybody else has questions. So I'm wondering, say I wanted to get an IFCB
Mhm.
and take it on my field runs, how sensitive would it be to transportation? Like if I wanted to just like bop around and do some discrete sampling,
Mhm.
do you think that would harm the mechanics of it to be moved around that much?
Depends on what kind of vessel you're in. So a lot of these vessels where they are taking it, they're like larger vessels, so it's not like you're on a motorboat that's like, so that's probably
Hmm.
not a good situation for it. It can be sensitive to like a ton of vibration, so a lot of people when they take it even on a vessel, they'll place it on something that kind of helps insulate it from like really vibrating. Because, right, you're going to take a picture. So even with your iPhone, if you're like vibrating, you get a bad image. So it's kind of like a same principle applies. And also you don't want to knock it out of alignment. So we've had, some of these on piers when you have a big storm and something hits it, everything all of a sudden you're just taking like you're either not getting images, you're like I can talk to it but it's not taking images because everything got knocked out of alignment, so you got to bring it back and kind of refocus everything and take it back. So it depends on your exact boat situation and the type of activity, but if you're like skipping along, that's probably not an ideal situation. You might want to like take your samples back and run it like in a benchtop mode or something like that.
Hmm.
um,
What do you think about like if I was like sampling, like if I'm driving around in my car and like stopping and shore sampling, if the movement of the car would be too much?
I don't know, like, it might be okay. It depends on what kind of driver you are, right? Are you taking the speed bumps really hard, or are you over them really
No
soft?
comment, Jamie. No comment. Let's see. So if we were to do like discrete sampling back at a
Mhm.
lab, would you need to like preserve it, like put some Lugol's in the sample, or how would that interfere with the pictures and IDs?
Yeah, that's a great question. So some people do that. Lugol's is hard because it changes, it can change the morphology and it can change the color, and it obviously darkens the actual sample itself. So some people have done it, and then there's fine tuning that you would want to do to so they get really they'll like change out the pumps, and they're like, these are my Lugol pumps, and these are my non-Lugol pumps. So there's just any if any of you have like run a flow cytometer, it's got the same like little they like life to be a certain way. And when you start doing different stuff, it's like you either need a different set and then you can't, you have your fresh set and your live your dead set of tubing and pumps. So you can preserve it. It's just a lot of extra like you'd probably want to just test it out and fine tuning, but people have done that.
Daphne?
Sorry, I'm trying to. It gave me a bunch of error messages when I was
Yeah.
trying to turn on my
Yeah,
camera. Hi, thanks for your presentation, Jamie.
sure.
This is I am just kind of a hanger on here. I work with Emily and others who are way more have way more expertise than I do, but
Sure, sure.
we are interested in an IFCB, and I'm wondering, it's kind of along the same lines as what Emily was talking about. If we're collecting samples in the field and we need to bring them back to the lab and we're running the IFCB in benchtop mode. How long, first of all, how long do you think, what timing do we have between like sample collection and then when it needs to be run in benchtop
Sure.
mode in the lab?
Yeah.
And
Yeah.
then how long would it take to run in benchtop mode in the lab, like a per sample? And I know it probably depends on like, well, how much volume did you collect and like what's in the sample and that kind of thing. But just kind of a
Yeah.
ballpark would be helpful.
Yeah. Yeah. Happy to so give you some what we've done, right, on the marine end is so on we have one that we run on benchtop mode periodically, depending on so you'd want to tune the instrument to how dense your sample is. So we run marine samples, which are often it's funny, like marine people are like, oh my God, it's the densest bloom. I'm like, have you ever been to Clear Lake? Because like you don't know what you're talking about. But we're talking about like in a marine side, like a dense sample is often a chlorophyll of maybe like 10. So so it's you're probably talking like two orders of magnitude higher than that sometimes, right? Or one order of magnitude higher than that in a freshwater system. So we draw the full 5 milliliters to get a representative sample at that kind of biomass level. And so at a 5 mil run, that takes about 25 minutes to run the sample. As far as holding time, we did some trials and have done stuff where we like put the cells in the refrigerator overnight and run it the next day. They usually look okay. You maybe lose some stuff, but like, a lot of people do that with microscopy. So it's like you're probably like same difference on that front. And so we try to run it within 24 hours of collection. So you can do the math there of like if you're going to run the full 5 mils, if you have a really dense sample, maybe you could cut the syringe pull to like 2.5, and then that would cut your run time a little bit. Um, and I would have to do the math on the top of my head, which I'm not able to do, but I think that would get you down to like 18 or 15 minutes if you cut the volume in half. Don't quote me on that, but that's like I think that's about where it is. But that the syringe pull is really what most of the steps are like a set time, and then how much volume you pull is really what shortens or lengthens your run. I think I addressed all your questions there.
Yes, yes, you did. I have
Okay,
follow up, but I'm going to wait.
sure.
I see others.
Yeah. So it looks like Nick is next.
Hi, yeah, thanks
Yeah.
for the great talk, Jamie. I was interested in like how much fine tuning does it generally take? Like if you're going to run this in a flow through system on a boat or on a pier or something, and you have like sure you have differing lengths of tubing leading from like the actual outside water to the IFCB. Um, is that like something that you have to control for in these deployments?
Yeah, that's a great question. You do, especially if you're sipping and you want to geolocate. So if you're in on a cruise and you're sipping and you have maybe variable lengths of tubing and you want to know kind of roughly where you got the sample, you do have to kind of know, okay, where did I pull from?
Right.
How long did it take to come through the tubing? To then kind of back calculate like the exact location. On a pier, like you know where you are, and maybe you're okay with a little bit of a fudge factor. A lot of times though, so the instrument itself can't pull from, so at some of these pier locations where it's in a on a top side, but in a controlled like kind of environment, it's pulling from, you know, anywhere from 5 to 20 feet in the air. So the instrument itself is not able to do that if it's that far down. So then there's a secondary pump system where you pump the water up into like a reservoir, and then the instrument will like sip from the reservoir, if that makes sense.
Yeah, that's
Yeah,
cool.
yeah,
Thanks.
yeah. Let's see, Ted.
I- One of the things that EMP comes up with, and that Tiffany and I have talked about over the years, is we because we use our um, we use the ship's onboard pump to
Mhm.
take our phytoplankton samples, and
Yeah.
sometimes we'll notice, especially with diatoms, that leads to like breakage, and
Hmm.
so I was wondering with the IFCB how I'm guessing the pumps are maybe a little more gentle, but I was wondering if you had experience with that, if you'd noticed that it causes cell damage or other things, especially
Yeah.
with more delicate kind of
Yeah,
organisms.
with more delicate. So for yes, I think the answer is yes. So depending on where and how the instrument is deployed, I've noticed this is totally anecdotal, but pseudonuclea can make really long chains with like a dozen plus cells. I feel like the images that I see, and this is also a function of the field of view of where it's taking the image, right? So you don't necessarily going to fit the whole chain. Um, in the field of view, but I feel like I often see that they're fragmented. And I feel like they're fragmented more in some of the stations where it's a pump that's pulling it up the pier into a reservoir. It's just been through a lot at that point. So I think it would be similar probably to what you're seeing with the inflow system is if, depending on how you deploy it, you are aware of that and you're like, okay, I'm seeing fragments. Also keeping in mind that you can't fit the whole chain in the field of view sometimes. So you're like, I see a maximum of maybe like 5 cells anyway, and I know from microscopy that they can get longer than that when you can kind of look like with a dissecting scope. But you do get some fragmentation. And so I don't have a ton of experience running this with in a freshwater context where, you have something like a microcystis that's in a colonial form. I couldn't speak to from my own personal experience like how that would break up a colony or not. So I would trust your judgment as far as like if you're seeing it shredded in a boat flow through system, it might also get shredded. The other thing that is the sipper. The sipper does have a guard on it because you want to protect the instrument and you don't want to have stuff that's going to plug up your plumbing. So if you have really large colonies, they might not actually go through the guard. The guard's like I think it's 150 microns, um, just to protect the internal plumbing.
Thank you. That's super helpful.
Yeah, yeah. Daphne.
Thanks.
Yeah.
I was wondering, so as we think about using an IFCB in a more freshwater system, are there any, so the idea of kind of creating references and getting all of these images, so we don't have a network really to draw from. If we were to do it, we'd be kind of the first movers.
Yeah.
And so I'm a little worried about kind of how much work that would entail and if there are any existing resources maybe that we could draw from, or if it would be kind of us standing it up by ourselves, or I'm sure there's some overlap with some of the marine species and
Yeah.
marine sources, but yeah, I'm just wondering if you have kind of a thought on that. And yeah, I know it sounds like creating kind of a reference and creating all and your image library can be pretty daunting.
Yes, the image library is daunting. I know there's a few places that have used the IFCB. So what you would want to do if you were gonna look into this. And again, I can speak from like kind of the marine side. I know that some of the image libraries, some of the ICBs have been run in SF Bay, so there might be some image libraries that already have microcystis in it, or some of the like delicospermum or some of the other things that you see in the delta. Um, that would be the folks that run up in the SF Bay to ask them. Because what you would want is you can't just take images from anywhere. They need to be IFCB images. Um, so that's the bummer where you're like, oh, there's like thousands of great pictures of microcystis, but that's not what the neural network is gonna kind of use. So you need images that are collected using the instrument. So I think that you wouldn't be starting from absolute zero, but I will say that it was several years of effort, partly because it was people doing it on the side, kind of like, oh, one can only sit and identify pictures of phytoplankton for so long before they're like, my eyeballs are gonna fall out. I need to take a break. That was part of it too, because it is a little tedious when you're like, oh, we were doing like a single run. So it was like, okay, we're gonna sit down and do like one syringe pull, and depending on the day, that was like, oh, this is like 2 hours of putting images into bins. And there's the nice thing is again, there's infrastructure that's in place that you could leverage. So there's nice programs where you can be like, oh, click, and it like automatically it takes some of the work out of like classifying, because they have a essentially a program that helps you classify by like showing you a couple images at a time. And so there's some of that stuff. So you wouldn't be like literally from zero. You could leverage some of that, but I would say I would assume depending on your system, there might need to be some image library building for freshwater.
Sounds good. Thank you.
Yeah, yeah.
You had mentioned a multi-day training course. Is that something that the manufacturer puts on?
Yes. Yeah. They
Okay.
provide, yeah. This is an instrument that's built by McLean. I should have said that. But they provide really great support. So, for example, I've been on the phone on like Zoom with Tom many times being like, I need to replace this part and I kind of forgot how to do this. And they're like, okay, take your screwdriver, go right there, like turn the camera so I can see what you're doing. They're in Massachusetts, so it's just the time difference. But they're great and they provide like a ton of support, like well after you purchase the instrument. So
That's definitely a bonus.
yeah.
Does anybody else have any more questions? Great. Well,
Cool.
thanks a lot, Jamie. That was super
Yeah.
interesting. I think we've got a lot of ideas here.
Yeah. Yep, they're fun, but there is a lot that goes into getting them to work. But happy to answer any questions if they come up after later on. But yeah.
Thanks. I
Yeah,
appreciate it.
cool.
Okay, well, we can go ahead and end 9 minutes early. Yeah, thanks a lot, everyone, for joining, and we'll send out the notes and recording afterwards.
Great. Thanks, everyone.